very much, Andrew. So, hi everybody, and thank you for joining our webinar today. It's great to have so many of you keen to hear about RapidFACT, a subject about which I'm personally very passionate. RapidFACT stands for Rapid Formulation Development and Clinical Testing. It's an approach that we launched in 2009 to respond to what we saw as suboptimal conventional formulation development processes. Since that time, RapidFACT has been used to screen over 150 formulations to increase exposure or modify the shape of the exposure profile in over 50 early development and lifecycle management programs. The goal of today's presentation is to provide an introduction to RapidFACT, illustrate the technique using case studies, and discuss developments made in the last 12 months. We will also have an opportunity to hear from Magnus Ronn, as a former sponsor of a RapidFACT program, which I hope will provide an independent perspective for you. In this presentation then, we will provide an introduction to our key delivery platform, Translational Pharmaceutics, and an overview of RapidFACT concepts, before handing over to Magnus to discuss the program that Tetraphase performed for oral formulations of their molecule, Pharmaceuticals. I'll then discuss our wider experience with RapidFACT, supported by two further case studies, including an overview of the breadth of formulation design spaces applied to date. Finally, I'll show an example of where we used emerging data from a RapidFACT trial to support development of in vitro and vivo correlations to enable downstream development. Before I begin, I'd like to first consider how our industry is structured and what that means for development. When you look at outsourcing organizations, there are groups who are configured to produce API, develop formulations, and manufacture clinical trial supplies, and they're integrated very vertically. However, these companies are very separate to the CRO test organizations, which exist in another channel. What this means is it takes a long time to transition a drug product from the make side to the test side of the integrated supply chains. The impact of this is to drive up the CMC data package required. It increases API consumption. Of course, it brings with that an associated cost and has further impacts on development time. If we now think about what this structure means for formulation development, we find that the process described on this slide has become established. A pharmaceutical development program is conducted, which will yield formulation prototypes to be screened through preclinical species, which is usually dog. Successful formulations from this step will then be subjected to conventional CMC processes, including product scale-up and ICH stability studies, to support regulatory assessment for a clinical study. This process takes a long time and carries considerable cost. Independent assessments of this are in the region of 12 to 18 months and $1.5 to $2.5 million in outsourcing costs. As a result of this time and cost, a lot of emphasis is placed upon laboratory and preclinical experiments. However, these are widely accepted as not being predictive of human bioavailability. And this in turn increases the risk of having to repeat the formulation development cycle, because the correct product is not identified within one cycle of this development process. At the heart of this problem is the fact that our supply chains are only vertically integrated, and that makes entry into clinical administration very challenging. What if we could integrate these channels horizontally? And this is what we've done at Quotient. At our facility in the United Kingdom, we have an 85-bed clinic on the ground floor of our unit, and right upstairs from that is a fully functional laboratory to pilot scale GMP plant. Right across the street we have pharmaceutical analysis and formulation development functions, and associated with the whole organization is a regulatory affairs unit, a project management structure, and of course data management and other associated clinical support functions. The horizontal integration of the Translational Pharmaceutics platform means that we can make a batch upstairs move it downstairs to the clinic and dose it into the patients, in some cases in less than 24 hours. Therefore, the furthest a product really needs to travel between production and dosing is no more than 200 feet. Because of this integration, the Translational Pharmaceutical platform allows us to enter clinical dosing with a shorter shelf life on our drug products, and therefore we can enter clinical with a reduced CMC data package. We would typically submit around 7 days worth of real-time stability data to support an equivalent shelf life. This has the impact of removing ICH stability studies from the critical path, which can instead be run in parallel with clinical work. As manufacturing is at small scale, development of pilot-scale manufacturing processes can be removed from the critical path to obtaining clinical data. We will only need to manufacture the drug product we need to support dosing. And finally, because cycle times between manufacturing and dosing are so short, we can allow rapid make and test cycles to come into the study design. This means that real-time decisions can be made on the basis of emerging safety, PK, and PD data, and that decision can influence the drug product that is manufactured and dosed in the subsequent dosing period. This will typically happen on a 7 to 14-day cycle time, really allowing this process to occur without significantly extending the clinical phase of the trial.